We found a whisper.
Welcome to Base by Base, the papercast that brings genomics to you wherever you are. Thanks for listening, and don't forget to follow and rate us in your podcast app. Imagine, just for a second, handing a pharmaceutical company $2 billion and like 15 years of your life, only to flip a coin at the finish line to see if the medicine they created actually works.
Yeah, it sounds absurd, but that is the terrifying reality of modern drug development.
Right, it's just a staggering problem. And honestly, that coin flip analogy is barely an exaggeration.
Barely. Because even after over a decade of research, all those billions invested, nearly 50% of drugs fail in the late stages of clinical trials. And it's simply because they lack efficacy. They get into the human body and they do not perform the biological task they were designed for.
Which is wild. And then what, another 25% fail because of unforeseen safety concerns?
Exactly, safety or toxicities that we just didn't see coming in the animal models.
So we have a system where basically 3/4 of these massive medical investments evaporate right before reaching the patients who actually need them. So the question is, how could this massive failure rate change if we could read the exact instruction manual of human disease before we ever even tried to design a drug?
Well, that potential to front load our understanding, that is the absolute promise of modern genomics. We are moving past just identifying that a disease risk exists somewhere in your DNA.
Right, like just knowing you have a risk.
Yeah, and we're moving to actually decoding the cellular mechanics of how it operates. And today we celebrate the work of the team at the University of Cambridge, along with researchers from Harvard and the Baker Heart and Diabetes Institute, who have advanced our understanding of multi-scale genomics.
It's really hard to believe it has been nearly 25 years since the initial draft of the human genome.
I know, time flies.
It really does. And for the past 2 decades, the heavy lifter in this field has been genome-wide association studies, or GWAS. And from my reading, GWAS was incredible at feeding genetic loci, right? Like these specific zip codes in our DNA that are linked to diseases.
It was a massive breakthrough, yeah.
But we seem to hit this massive scientific bottleneck. Like we found the statistical associations, but we were completely missing the biological mechanism.
Right, and that bottleneck has been really the defining frustration of genomics for years. GWAS gave us this massive list of genetic coordinates. It allowed us to say, that people with a variation at coordinate X have a higher risk of heart disease.
But just having the coordinate doesn't really help you fix it.
Exactly. A coordinate on a map doesn't explain the biology. It doesn't tell you the how or the why. And without understanding the actual mechanical breakdown happening inside the body, trying to design a drug to fix it it's essentially guesswork.
If I'm understanding this correctly, it's kind of like GWAS is basically a satellite map showing a traffic jam in a city.
Okay, I like that.
Right, like it tells you exactly where the congestion is, but from space, you have no idea why the cars are actually stopped. To figure out if it's, a pothole, a broken traffic light, or a parade, you have to zoom all the way down to the street level.
The street level view is a really great way to think about it. And achieving that view is the entire premise of this new multi-scale genomics. We have to move away from just, population-level statistics and actually integrate data across 3 distinct biological scales.
Which are the molecular level, the cellular level, and the whole organ level, right?
Yes, exactly.
So let's talk about how researchers actually pull off that zooming process. Because when we look at the cellular level, the sources talk heavily about single-cell RNA sequencing.
Yes, cRNA-seq. It's revolutionary.
Because our bodies aren't just one uniform block of DNA, right?
Yeah.
We have skin cells, neurons, heart muscle cells.
They all share the exact same genome.
Right, but they operate completely differently. So this sequencing tech, it lets you see which specific genes are turned on or off in a single specific cell at any given moment.
Exactly. It provides the immediate genetic context. But, just observing that a gene is turned on doesn't actually prove that a specific genetic variant is controlling it.
Oh, interesting. So how do you prove that?
Well, to bridge that gap, researchers use something called expression quantitative trait loci mapping.
Okay, EQTL mapping.
Right, EQTL mapping.
Yeah.
This technique links a genetic variant directly to the regulation of a gene within that specific cell type. So it proves a causal chain of command.
Okay, so that handles the cells and the molecules, but scaling up to the whole organ level, that seems vastly more complicated.
It is. It requires a massive leap in data integration, which brings us to imaging genetics. Researchers are tapping into these population-scale databases, most notably the UK Biobank.
Oh, I've heard of them. They just hit a big milestone, right?
They did. They recently crossed a milestone of 100,000 MRI and PE scans of the brain, body, and bones from volunteers.
Wow. 100,000.
Yeah, it's a gold mine. And they feed this visual data into deep learning models to extract high-dimensional data points, which they call imaging-derived phenotypes, or IDPs.
Wait, I want to make sure I'm grasping this. They have an AI analyzing tens of thousands of medical scans, mapping out the microscopic physical shapes and structures of these organs, and then cross-referencing those physical shapes with the patient's genetic codes.
That is the foundation of it, yes. But, I mean, generating the data is really only half the battle. The real innovation is the modeling used to interpret all of it.
Yeah.
They rely on these frameworks like stratified linkage disequilibrium score regression.
Okay, stop right there. Stratified linkage disequilibrium score regression. That sounds like someone dropped a dictionary into a blender.
I know, scientists are terrible at naming things.
What is that actually doing in plain English?
Fair enough. Think of it as an incredibly sophisticated calculator. It doesn't just find a broken gene, it calculates exactly how much that specific genetic variation contributes to a physical, structural change in a specific cell or organ. Right, so it measures the heritability of a disease and maps it directly onto the physical architecture of the body.
Which brings us back to those failing drug trials we started with. If we can map the exact architecture of a disease, it makes perfect sense that we can figure out which drugs are actually worth developing.
Exactly. The pharmaceutical industry calls this therapeutic target prioritization, and honestly, the numbers are definitive.
Oh, definitive.
Well, therapeutic targets supported by human genetic information are 2.6 times more likely to succeed in clinical trials.
2.6 times.
Yeah. And that single metric represents billions of dollars in years of saved time.
I'd love to ground this in a concrete example. Like, how does knowing the genetic architecture translate into a physical drug on a pharmacy shelf?
One of the most elegant examples of this comes from studying rare genetic mutations. Years ago, researchers noticed that a specific subset of people of African descent had unusually low cholesterol levels.
Okay.
And they had almost zero risk of cardiovascular disease. It turned out they carried a rare natural loss of function mutation in a gene called PCSK9.
So nature basically ran a biological experiment showing that if this specific gene is broken, people don't get high cholesterol.
Exactly.
But how does that actually work mechanically? Like, what is PCSK9 doing that makes breaking it a good thing?
The mechanism is really fascinating. In a normal system, the liver had these LDL receptors on its surface. Think of them like tiny vacuums pulling bad cholesterol out of the blood.
Okay, vacuums. Got it.
The PCSK9 protein's job is to bind to those vacuums and destroy them, so the liver doesn't clear too much cholesterol. But if you have this rare mutation, you don't produce the PCSK9 destroyer protein.
Which means the liver gets to keep all of its cholesterol vacuums. They just stay on the surface of the cell, constantly pulling bad cholesterol out of the bloodstream. So pharmaceutical companies just had to design a drug that mimics that broken gene, right? A drug that blocks PCSK9.
And that is exactly what happened. We now have FDA-approved PCSK9 inhibitors that are absolute blockbuster drugs for people who suffer from severe cardiovascular disease.
That is incredible. And there are other examples, right?
Many. We saw a similar pathway with the TYK2 gene, which was prioritized as a target for autoimmune diseases and led to a new oral medication for psoriasis.
Wow.
And of course, the BCL11A gene. Genetic studies showed it suppresses fetal hemoglobin, which led directly to the development of Casgevy.
Wait, Casgevy? That's the
world's first approved medicine based on CRISPR gene editing for sickle cell anemia, yes.
That's huge. But this multi-scale genomics approach isn't just about ensuring a drug works, is it? It's also about predicting whether a drug is going to be toxic to you before you ever even swallow the pill.
Yes, exactly. The field of pharmacogenomics is becoming remarkably precise. Severe adverse drug reactions, which account for a massive portion of those clinical trial failures we talked about, are often driven by human leukocyte antigen or HLA variants.
Right, like if a patient carries the HLA B57.01 variant, giving them a common antibiotic like flucloxacillin can trigger severe liver injury.
Exactly. And knowing that genetic quirk exists allows doctors to simply choose a different antibiotic and completely avoid a life-threatening reaction.
Okay, so if we're talking about these genetic quirks and broken genes, I want to pivot to something from the sources that completely reframed my understanding of DNA.
Oh, What's that?
Well, I always assumed that the genetic variants causing disease were sitting inside the genes that actually build our proteins. But most of the variants identified by GWAS don't sit in the coding regions at all.
Right.
They sit in the non-coding regions, like the vast stretches of DNA we used to dismiss as junk.
Yes. Unlocking the non-coding genome has been one of the great aha moments of the last decade. We knew these non-coding variants were strongly associated with diseases, but because they didn't actively manufacture proteins, they were a complete mystery.
But now we know what they do.
We do. By integrating the single-cell data we discussed earlier, we can finally see what they're doing. They are regulatory elements.
It makes me think of like a movie set.
Okay, how so?
Well, for decades, we were so obsessed with the coding genes, right? The actors reading the script on camera, that we completely ignored the non-coding DNA. But those non-coding regions are actually the directors.
That's a great way to look at it.
They're standing off camera, telling the actors to be louder, to be quieter, or to stop talking entirely. And the single-cell data finally lets us hear the director's instructions. But we're learning they only give those instructions in very specific cell types.
The director analogy captures it perfectly, especially when we look at complex conditions like obesity. Because for a long time, the assumption was that the genetics of obesity were tied to adipose tissue.
Meaning that fat cells themselves were genetically predisposed to store more energy.
Right. But the data tells a completely different story. When researchers took a GWA for body mass index and mapped it against this new spatiocellular map, they found that the genetic variants driving obesity aren't enriched in fat cells at all.
Where are they?
They are heavily enriched in mid-hypothalamic neurons.
In the brain.
In the brain. The implications of that discovery are profound. It proves that genetically, obesity is fundamentally a neurological and behavioral condition. It is an energy balance equation dictated by brain signaling.
The brain is telling the body it is starving when it actually isn't.
Exactly.
Which entirely shifts how we view the condition. And incidentally, it completely validates the mechanism behind drugs like Ozempic and Wegovy.
Absolutely it does.
Because those GLP-1 agonists don't burn fat directly. They act on the receptors in the brain to regulate satiety. The genetic map finally proved why targeting the brain is the most effective way to treat weight loss.
And we are seeing similar paradigm shifts in neurodegenerative diseases too.
Like Alzheimer's.
Yes. For years, Alzheimer's was viewed primarily through the lens of neuronal degradation, just brain cells breaking down. But multi-scale genomics revealed that Alzheimer's risk is actually heavily mediated by microglia.
Microglia.
Yeah, which are the primary immune cells of the brain. They act like the brain's trash collectors.
So if the genetic directors in the microglia give the wrong instructions, the trash collectors strike and those beta amyloid plaques just build up.
Exactly. It reframes Alzheimer's from a purely structural breakdown to an immune inflammatory failure.
So, okay, if these non-coding directors are altering how single cells like neurons or microglia behave, how does that collective cellular chaos physically reshape an entire organ?
Well, that is where the imaging data from the UK Biobank becomes so critical. Researchers can now link specific genetic variants directly to macroscopic changes in the body.
Like what kind of changes?
Like, they can track the genetic loci responsible for thickening the regional walls of the heart's ventricles. In the brain, they can map the specific variants that accelerate biological aging or drive the physical accumulation of those amyloid plaques we just mentioned.
This raises a massive question for me, actually, because I was looking at the findings on multi-organ connections in the sources.
Yeah, that's a wild area of research.
It is. The researchers discovered that the fractal dimensions of cardiac trabeculae, which is essentially the complex, spongy scaffolding on the inside of the heart muscle,
Right.
they share the exact same genetic loci with the dendritic complexity in the brain.
They do.
If I'm a patient listening to this and I realize my heart's physical structure shares a genetic blueprint with my brain's physical structure, I'm suddenly wondering if a therapy designed to remodel my failing heart might accidentally alter the wiring in my brain.
It is a critical concern, and it strikes at the heart of what biologists call pleiotropy.
Pleiotropy.
Yeah, pleiotropy occurs when a single gene influences multiple, seemingly unrelated physical traits. The challenge for researchers is disentangling that shared blueprint from an actual causal interaction. Like, are the heart and brain just built using the same genetic toolkit? Or does a struggling heart actually cause the brain to degrade?
How do you even begin to separate those two things? You can't just run a clinical trial where you intentionally induce heart failure to see what happens to the brain.
No, you definitely cannot do it clinically. but you can do it genetically using a technique called Mendelian randomization.
Okay, how does that work?
It essentially uses your genetics as a natural randomized controlled trial. Because your genes are assigned randomly at conception, researchers can group people by genetic risk, completely bypassing confounding lifestyle variables.
Oh, that's clever.
Very. And what they're finding is it is often a combination of both. There is pleiotropy, but adverse cardiac remodeling also causally accelerates neurodegenerative conditions.
Wow. Okay, so if they've isolated the malfunctioning heart muscle cell, I'm assuming the next step isn't just throwing drugs at it blindly. They must be looking for the specific gears inside that cell that are broken.
Exactly.
Let's walk through the case study on heart failure to see how all these scales integrate.
Sure. So the multi-ancestry GWAS for heart failure gave them the initial genetic coordinates. Then, layering on the single nucleus RNA sequencing revealed that the risk was enriched specifically in cardiomyocytes.
Which are the heart muscle cells.
Yes. So once they knew the exact cell, they moved down to the molecular scale. They conducted a proteome-wide analysis connected to those cellular signals and identified 9 specific circulating proteins in the blood that are causally linked to heart failure.
9 proteins. It went from a vague statistical association across millions of people down to 9 specific molecules.
Incredible, isn't it? And those 9 proteins instantly become prioritized, genetically validated targets for a new heart failure drug.
And looking at where this field is heading next, it feels like we are crossing into science fiction. The paper discusses an explosion of AI foundation models, similar to large language models, but built for biology.
Right, tools like SCGPT for single-cell data or ProtST for protein sequences.
Yeah. And how does that change things?
The integration of these AI models with multi-scale genomics is driving just a breathtaking technological leap. We are moving toward a reality where researchers utilize techniques like PerturbSeq.
PerturbSeq. The sources mention that combines large-scale CRISPR genetic screening with single-cell transcriptomics.
Yes.
Wait, let me make sure I understand PerturbSeq. You're saying we can use CRISPR as a giant biological stress test. We can intentionally break or mutate thousands of genes at once in a petri dish, and then use AI to watch exactly how the cell panics and responds.
That is the perfect way to describe it. You are intentionally stressing the system to map its ultimate breaking points. And doing this at scale is leading to the development of virtual cells.
Virtual cell.
Yeah, by feeding all this multimodal data into foundation models, we can simulate human biology on a computer. We will soon be able to predict how a specific cell will respond to a novel drug in silico.
In silico. So before a single chemical is synthesized in a lab, let alone tested in a human patient.
Exactly.
As incredible as predicting drug responses in a virtual cell sounds, we need to ground this. Every major scientific leap has a catch. What are the limitations holding this back?
The limitations are significant. First is the black box nature of these advanced AI models.
Meaning we don't know how they get their answer.
Right. When an algorithm extracts a complex feature from a heart scan and predicts heart failure, it cannot always explain what that feature is in human biological terms. If a cardiologist cannot understand the physiological reason behind the AI's prediction, well, it is incredibly difficult to justify using it in clinical practice.
That makes sense. And beyond the algorithmic black box, there is the fundamental issue of the genetic data itself. The sources made it very clear that our genomic databases are overwhelmingly skewed.
It is arguably the most critical bottleneck right now. To perform the fine mapping required to identify true causal genetic variants, researchers desperately need to expand ancestral diversity in these massive studies.
Especially African populations, right?
Yes. Currently, there is a severe lack of data from African populations in particular. Human genetics is incredibly complex and diverse. If the foundational databases we use to train these virtual cells do not reflect global genetic diversity, the precision medicines we develop from them will not either. It is a mathematical and biological necessity to fix this imbalance.
So, synthesizing everything we've covered today, genomics is no longer just about handing a patient a vague disease risk score based on a statistical map. It has evolved into a multidimensional blueprint that connects molecular pathways to specific cellular behaviors and ultimately to the physical physiology of our organs. By translating these genetic signals across all of these scales, we are fundamentally de-risking the entire drug development pipeline and accelerating the arrival of true precision therapies.
What does this mean for the future of medicine? When your specific genetic blueprint dictates not just your risk of illness, but the precise cellular mechanism and physical organ structure a new drug will target.
It is a complete paradigm shift for how we treat human disease. And before we wrap up, there is one final concept from the research that I haven't been able to stop thinking about. The researchers discussed the idea of genetic aging clocks across different organs.
Oh, that's fascinating.
Right. It raises a crazy question. Could your unique genetic blueprint dictate that your heart is biologically aging 10 years faster than your brain? And if multi-scale genomics maps those clocks perfectly, could we eventually prescribe targeted drugs that reverse the biological age of one specific organ at a time? It fundamentally alters how we think about the aging process itself.
It really does.
This episode was based on an open access article under the CC-BY 4.0 license. You can find a direct link to the paper and the license in our episode description. If you enjoyed this, follow or subscribe in your podcast app and leave a 5-star rating. If you'd like to support our work, use the donation link in the description. Now stay with us for an original track created especially for this episode and inspired by the article you've just heard about. Thanks for listening, and join us next time as we explore more science base by base.
We found a whisper in the noise on the screen, a tiny variant where the truth might have been. Not just a dot on a Manhattan sky, but a door to the why, behind the why. So we zoom in, line by line, getting closer. From the map to the gene to the cell that holds it. Crossing layers, making patterns align. To the hidden hand finally shows its design. From signal to cure, let it light up the night. From code to the clinic, we're turning it right. Find the target, name the spark, make it real. One more step from what we guess to what we hear. To what we hear. Of the tail, Mendelian bets that can sometimes prevail, and images of organ structure, motion, strain, linking what we carry to what we can change. From signal to cure, let it light up the night. From code to the clinic, we're turning it right. Find the target, move the odds, make it real. One more step from what we guess to what we hear.